There is one single thing that connects you to a blue whale, a giant redwood tree, and a mushroom, and is invisible to naked eye. It is the cell, the ultimate blueprint of all living things. Hi, welcome to Tehsin's Life Sciences. Let's dive to introduction about cell. Cell, what is the definition of the cell? Cell is the basic unit of life, the unit of all living organisms. Taken an example of a building made of bricks. In case of building, bricks are basic unit of this building. This is a typical diagram of an animal cell. Next slide is about what is section. With the help of a razor blade, when you cut a small piece of a plant or an animal tissue, then this small piece you have cut is called section. What will you do with this section? You just observe this small piece of plant, either plant tissue or either animal tissue under the microscope to observe its structures. Okay, then there are two ways to cut this section with the help of razor blade. You can cut either along the length or along the diameter. And which you cut along the length, it is called longitudinal section, and which you cut along the diameter is transverse cross section. We will study about plant cells. Okay, mostly plant cells are shaped like box shape. Here you are seeing a stem of plant, and there are many box-like cells are here. Okay, this is the diagram that has been taken, or the picture taken under to a 600x magnification. Okay, let's study about some characters of plant cells. As you know, the plant cells are made up of cell wall. In case of cell wall, but what about cell wall? Cell wall is the outer covering present in the plant cells. But in case of animal tissue, the cell wall is absent. And this cell wall, it is because made up of a cellulose. So it is very rigid, or it gives its strength to plant cell, making it is very easy to just cut with the help of a razor blade. So there is another thing that it is easy to cut. There is another thing is like the cells are large in size. So you can easily observe them under 600x magnification of microscope, right? But in case of animal tissue, you are seeing here the diagram, their shape is mostly spherical. Okay, animal cells are typically, they don't have cell wall, but they do have cell membrane. In plant cells, they have extra protective layer is that is cell wall, but they also have cell membrane. But in case of animal tissue, they have only have cell membrane as a protective covering. To cut an animal tissue, because they are very flexible and their size is very small, slippery, so we it is not easy with the plant cell. Animal cells are not easy to cut. So we will study this technique in the next slides. You just memorize that they don't have cell wall, their size is small, so you have to observe on at least 700x magnification under microscope, and their shape is not box-like, it is mostly spherical. Next slide is about staining. What is the purpose of staining? We use stains for clarity of this slide. You are here. There is a comparison about two slides, in which in one slide we have not used any stain, and the one slide we have used stains. Okay, and you can easily observe that the slide in which we have not used stains, it is very blurry, and the cells are just transparent and black and white background, and we don't have any visibility of nucleus or walls, not very much contrast. But in case of extent slide, there are very beautiful cells that are easily visible. Okay. Now next question is about which stains we can use. There are two types of stains. We can use methylene, and there is another is iodine. The color of methylene stain is blue, but in case of iodine, its color is golden brown. And iodine is mostly used for cell wall. Iodine is used to color starches or carbohydrate parts or cell wall. but within mostly colored nucleus. So in this diagram, the blue part is nuclei and the outer layers are mostly membranes, cell wall or the starch part. Next topic is how to prepare the slide for plant tissue to observe under microscope. You just have to take a plant or an onion, just to cut into four pieces. Then take one piece and further taken this scale, just taken this leg, you have to break it. After breaking this scale of this onion, you just have to peel its transparent like epidermis layer. This by after taking this epidermis layer, you just have to cut into small pieces to fit on the slide. On the other hand, you have to take a slide where you have to place a drop of water, because if you not place a water or a drop of water on the slide, then the plant tissue will dry out. They will dehydrate it, and you can't shrink, and you can't observe them in their original shape. Next, you can also use a stain. Here is a diagram of methylene blue solution or a iodine solution, and then just place a cover slip. But you have to place cover slip on a 45-degree angle. After placing cover slide, you just observe under the microscope. Next slide is about how to prepare it for animal tissue. In the previous slides, I have already told you that that animal tissues are their size is very small. Their surface is flexible, slippery, floppy. You have to use a technique that is embedding. You have to take a wax. You have just take a container, and there is a wax. Place a tissue. When this molten wax, this is hot. This is liquid wax you have taken, and just place the tissue, and after just let it cool. After cooling, it becomes solidified. Now it is very easy to cut because it is trapped inside the solid, and you will cut with the help of a razor blade, or you can any use device. And then after sectioning, you can place them the slide and repeat the process and the staining and observe under microscope. If you take a look on this slide, there is a technique that plant epidermis peeling. We have done it with the onion previously, but here we are peeling epidermis from the plant leaf. For this purpose, you have to take a leaf, just just break it into two small pieces. After breaking a turnover of this leaf, you will see a transparent epidermis layer. Just put out and repeat the same process as you have done with the previous slide. There is another technique is cell smear preparation. Yeah, we use this technique for mostly blood. We just took a blood sample, and with the help of two slides, we just smear them, slide them on each other. In this way, the blood cells are become loosely bound, and you will easily observe it under the microscope. There is no single shape, no standard shape. There are many shapes and of the cell according to the function. Here in this diagram, you are seeing many types. First is the neuron, blood cell, muscle cell, macrophages. All these types are of cells, and they have their own function. Like the shape of the neuron is branch-like is because because the neuron has to transmit message or nerve impulses from one place to another. So it needs a structure that will be branching. But in case of a blood cell, because it has to move smoothly from the arteries or capillaries, so its shape should be round. So it is not considered that there is just a box-like shape or spherical shape. There may be many different shapes according to their function. Okay, last we will see mainly three components of cell that are very important. That is, first is cell membrane, then nucleus, and then cytoplasm. Cell membrane is like a gatekeeper of the cell, which there is a term selectical permeable. It means that it controls whether or which molecules can enter and which molecules can't. So every molecule needs its permission. Then there is a nucleus. Nucleus is like a brain of the cell. It controls all activities related to growth, related to genetic material, or which protein should be made or not, or which things are needed where and they should be transported, all controlled by nucleus. Next is the cytoplasm. Cytoplasm is jelly-like material, and it provides medium where the cells all activities will happen. It is like a housing society where all facilities are provided. You just have to build a house and just live there. So cytoplasm is like this housing society, or it is a jelly-like substance where all chemical reactions, where all organelles are present, and the cell will grow and do all its activities. There is a typical diagram of plant cell. This is the animal cell. In case of plant cell, you are seeing that its shape is box-like, and in the typical animal cell, the shape is spherical. And we will discuss all its components in further videos. Thank you. If you like this video, just subscribe and like and comment this video.